my voice?
Yes, we can.
Thank you. Yeah. I think we can begin. Dr. Khalipo, can we begin?
Yes, of course.
Thank you. Dear keynote speakers, dear guests, Tehran University of Medical Sciences and Farabi Hospital welcomes you to the Appraise to Raise webinar held by Deputy of International Affairs of Tehran University and Farabi Hospital. We would have 6 distinguished speakers, which all work as leaders on different aspects of image analysis around the world. I believe image analysis supposed to be the future of many parts of ophthalmic imaging. In this webinar, I think we can know that in future, image analysis helps novice clinicians diagnose the diseases as well as maybe most expert clinicians in that field, if not better. Our first speaker is Professor Tande Peto. I'm honored to introduce her. She is Professor of Clinical Ophthalmology in Queen's University of Belfast. She leads the Northern Ireland Diabetic Retinopathy Screening Program. She is also Director of a Central Administrative Research Facility, a unique facility for grading of ophthalmic imaging. She is one of the most influential researchers in diabetic retinopathy around the world, especially in complications of retinopathy. She will talk on ophthalmic imaging and image analysis techniques, especially for telemedicine. Dr. Peto, we will listen to your presentation.
Thank you very much. Let me we shared the screen before. Can you see my screen now?
Yes.
Brilliant. So thank you very much for the invitation, and I'm delighted to be speaking here. And I'm going to use diabetes and diabetic eye disease to illustrate some of the basic principles, and a lot of my speakers will be building on these principles. So as you know, diabetes is really a real issue, and especially in the Middle East and Caribbean, where in some countries up to 50% of people have diabetes. And a lot of these people will have diabetic retinopathy over time. When we have looked at how eye health contributes to sustainable development goals, it turns out that it pretty much helps with every single goal, and it helps us to be able to provide good quality education, well-being, and getting out of poverty. So it's very important that we deal with it. Unfortunately, diabetic retinopathy is the only kind of blindness and visual impairment that's still on the rise. And unfortunately, some of the patients are still very upset about their disease, and they will have poor physical and mental health if they have diabetic macular disease and diabetic retinopathy. And what is staggering from this report is 1 in 5 ophthalmologists around the globe have never received any training on the treatment of diabetic macular edema and diabetic retinopathy. But the good thing is that we need, while we need to put the eyes back into the body and be able to see the disease itself, everything in diabetes, or most things in diabetic eye disease, can be imaged. So we usually image the retinopathy, we image the maculopathy, we talk about cataract, But those of you who work in cornea and anterior segment, you will know that people with diabetes a lot of the time have real issues with their corneal nerves. They have repetitive injuries, and they then eventually, a lot of the patients, if they are untreated, they might go into rubiosis and rubiotic glaucoma, and some patients will need VR surgery. So we need to be able to image the eyes, and we need to be able to provide some good treatment for these patients. So when we imaged the eye in the UK, we started screening diabetic eye screening it back in 2002 based on a national protocol. And we photographed two areas of the eye, one centered on the disc and one centered on the fovea. And this covers about 18 to 20% of the back of the eye. And we make decisions based on this 18 to 20%. So if we see some areas of exudates next to the fovea, or we see beading as in the very bottom of the image, and lots of other changes, then we will be referring the patient based on those. Those who have no gradable image, they will go into a slit lamp pathway, and optometrists and now technicians who we have trained up to be seeing these patients, and sometimes doctors will be seeing these images. But in order to have this very good quality image analysis, we need to get very good quality images. So a lot of the times we need to dilate the pupil, and in some countries this might pose a real issue, because if everyone is driving, then you might not be able to dilate over the eyes. You need to make a decision as to which camera you want to buy, how many images you want to take. The more images you take, the longer the grading will take, the image analysis will take. And you need to make sure that those who are allowed to talk to the patients and do the image analysis, they are all quality assured. Once the patient is identified, then of course they need to be able to get some treatment, either laser treatment or injections, but we really do need some safe long-term treatment options without some of these very expensive options that a lot of people around the globe cannot afford. So what do we use all of these images for? We use them for streamlining the process to understand better who needs to be seen and stratifying our patients better. So on these two graphs that are from the English National Program, if you have a look at the bottom left of the graph, then you will see that if it starts at 0 time point, those patients who develop no diabetic retinopathy between the two screenings a year apart, here and there, their risk of having progressed to either proliferative disease or maculopathy is extremely low. And this is only for the two image sets, the two screening images that we take. However, if someone progresses in both eyes, then their risk of developing proliferative disease in 4 years is 1 in 3 and 1 in 4 for maculopathy. This means that those images are extremely crucial to be good quality in order for us to be able to pick up the change, the small change that might occur between no retinopathy and some retinopathy in both eyes. So what else do we need to use the image analysis for? This is one of my patients, and you can see that on the top that it's a fluorescein image. And of course, you would know that sometimes we don't do fluoresceins in every case, but in this case we did. And you can see the neovascularization in a couple of places. You can see that there is a lot of leakage on the fluorescein. And when you do the OCT, you can see that the patient has a lot of maculopathy. There's subretinal fluid as well as large areas of intraretinal fluid as well, and a very, very thickened macula of 929 micron. So we need to remember that when we start treating the patient, sometimes things go wrong even with treatment. So you can see that I've done all the laser PRP, and the neovascularization has fibrosed up beautifully. The macular edema has gone, but the retina has suffered as well. So looking at the layers of the retina is very important because the thickness is good, but there is the photoreceptors and the EZ line has gone. And sometimes we need to think, because we need to be able to see that this patient is not getting better even after the steroid treatment, and they got very, very poor visual outcomes as well. So if you look at some of these patients, then we need to think as to how we might be able to reclassify diabetic eye disease, because we know that 1 in 9 have more severe disease once the periphery is examined. So have we been missing things just on the 2 images? And unfortunately, we are missing some stuff. You can see it on this image set, same patient on the same day, minutes apart. On the conventional camera, you can barely see a few laser spots. Once you use the IDON, which is a confocal white light camera, you can see the new vessels that this patient is still active, and you can still see the new vessels on the Optos as well. So your grading might be different depending on which camera you use, and it's a very important lesson. You need to know your camera very well. So in the UK, we still don't have the OCT in the mandatory screening pathway. Patients get referred for OCT clinics because there's a lot of training, quality assurance, and a lot of cost issues if you want to have it for the nearly 5 million people that we need to see every year. And we have been working very closely with Optos and Optos Monaco, because this is a wide field image of Optos plus an OCT. So you really need to be, we really can minimize the patient time in clinic. During COVID, we were down to 4 to 6 minutes in clinic while the patients got undilated images, and then they went home, and then we would analyze the images later, and we would ring the patients later. So you can go really big like this, or you can go very small. And you are able to look at your areas of geographic area and see which one would be better for you, because we established that some of these handheld cameras are just as good as some of the big cameras if it's used well. And this was done in the Philippines. And of course, their geography of 7,461 islands lends itself for being able to carry the camera and the equipment with you. You can use some of the iPhone attachments as well, and some of them are getting really, really good. And then once you look at the images, some of these artificial intelligence softwares, they have now passed regulatory processes as well, and they have approval. But a lot of them required a lot of human grading. A lot of them required all the information that we had from the previous images as well, and we needed to be grading for gradability and also for the image sets that we have. So we are in the process of doing the AI in ultra wide field images and making sure that we don't miss anything, and it's becoming very, very good. So hopefully that will come soon enough. And just to finish off, the most important thing is that we look at very clearly at these images. We need to use all the tools that we have available to us. Otherwise, we might find that we're missing all of these abnormalities that you see on this image. And you make sure that we achieve what the patients want, which is good driving vision, stable and improved visual acuity, and gaining the letters back so they can do their normal life and they can go to the shops and they can come to the clinic unworried. Thank you very much for your attention.
Thank you very much, Professor Tandipetu, for your comprehensive and nice presentation. Thank you. Any of the audience, if you have any question, please raise your hand. If not, I will ask some questions from Dr. Petu. Is that possible?
Yes, of course.
One of the concerns about the enrollment of telemedicine platforms in clinical practice is security measures. What do you think about the security measures that can protect patient data and ensure the privacy of ophthalmic images shared through the telemedicine platforms?
So a lot of the countries have their own legal terms and the legal framework as to what is allowable, and that we definitely need to obey. We find that as long as we're working within the confines of the NHS, and we do not leave our laptops without password and open, and also make sure that we don't take anything home on the tube or on the bus. A lot of these things are great. What you always have security issues if you have everything in the cloud, but knowing your systems well is amazingly important. Also, to make sure that everyone is well trained, because it's quite easy to rush some of these image analysis, and then you end up pushing the wrong letter out or to the wrong patient, or you might have misread the abnormalities. So you need to security of the system is one thing, but security of good quality information, good quality grading is also part of the pathway that needs to be developed together with every other aspect of imaging and image analysis.
Thank you very much. And then my another question is, what advice would you give to healthcare professionals interested in incorporating ophthalmic image analysis into their telemedicine practice, and what resources are available to support them in this regard?
So that's a very good question. So there are quite a number of online courses, but a lot of them are in English. And so some countries might, or some people might suffer not being able to do it. There are regular meetings for diabetic eye disease, but I think the most important thing is that you incorporate it into your daily practice. So it's not good to have a very busy clinic or a very busy operating theater from 7 in the morning, 7 at night, and then you go home and you start reading images. You're going to make mistakes, and the patient might suffer, and their eyes and vision might suffer. So what we're trying to do here is that it is, virtual clinics and virtual reading is pretty much just as a recognized session as a normal clinical session. You might be making more decisions, but we do find that if you need to speak, if you have an Optos and an OCT and the clinical information, around 18 to 20 patients worth of data can be read very carefully. In screening setting, we read more, but for the high risk patients, that's about it. And if you also need to ring the patient and tell them the information and discuss further steps, you might be down to 12 to 15 patients a session. For us, a session is half a day.
Thank you.
Can I ask somehow related question to your question, Dr. Khalilpur? Can you hear my voice, Dr.
Yes,
Petrov?
Yes.
Professor Petrov, I just wanted to ask that as you mentioned, in telemedicine, we use image analysis and it's incorporated in our practice well. But I think in routine practice and daily practice of our clinics, we don't see any effect of image analysis. I wanted to ask you, do you think in future it will be, or have you incorporated it in your daily practice, except for telemedicine?
Yes, so we do. So all of our diabetic eye screening program is based on telemedicine principles. And also for clinical things, every clinic that can incorporate imaging and image analysis will do so. Then the benefits might be slightly hidden. for example, if I go on leave, my clinics will still go on, and the patients will be imaged, and then the images will be read. So nothing comes to a complete standstill. And then we're using a lot of the proprietary softwares, but as I said, AI will be coming, and we'll need to work out how to get those images read quicker but safely by some of those new softwares.
Thank you. Thank you very much for your very nice presentation. I just wanted to introduce the second speaker, Professor Imre Lengil. He's a professor of Queen's University, Belfast, where mostly he works on translational ophthalmology. His lab mainly hello, Professor Lengile. His lab mainly works on image analysis in different diseases, including multiple sclerosis, Alzheimer's, and age-related macular degeneration. He authors many articles in different high-quality journals. I think we have a recorded video of your presentation. Do you prefer to go live, or just we present the video? Or I think if you want first introduce the video, and after that we can play it.
Yeah, I thank you very much for the invitation, and I think it's preferable to have the video. My flights were changed, and I'm sitting at the airport, so there is quite a noisy airport. So thank you very much for the invitation, and I think I'm basically looking forward to any questions and any thoughts on the presentation. So I would like to ask you to go ahead and show the video.
Thank you.
Dear colleagues, thank you very much for the opportunity to talk to you today. And I decided to record my talk because I'm traveling in Germany, and sometimes train connections are not very good. So I didn't want to leave you in the cold water. So therefore, I'm recording this. I will be online, and I will be, hopefully, and I will be able to answer any questions. So what I would like to talk to you about is drusen, and you are all familiar with it. You can see it on OCT very well. The only difficulty is that we don't know how it's made and what its function is. And we also know that there are different forms. Some of them can be seen on this OCT image. One of the opportunities that I had in the past was working with clinicians and scientists to look at a particular type of drusen. This is a fundus photograph where you can see circled a group of drusen, which I would like to explain why that might be important to you. If you take a baseline image, you can see how they look like. When you take a final image at the end of the patient's life, you see that it slightly changed. The size and position didn't, but its appearance did. And this it really becomes interesting when you take the autofluorescence image. At the baseline, you can see that there are an autofluorescence change in activity, but at the final stage, all those RPE cells die. So these drusen cause atrophy. This is obviously not good, so it's very important to understand how these atrophy develop. For this, we used the multimodal imaging approach, whereby we took images with OCT, color fundus, fundus autofluorescence, and near infrared in the same time on the same set of drusen from the same patient. And what you see is different features. So why these looks like, which the arrows point to as drusen, they have a hyporeflective core. They are non-reflective, originally on color fundus. They have an autofluorescence. They do produce this atrophy, but they also have a reflection in near infrared. There is another feature inside, that little dot in the middle, which we call hyperreflective dot. These hyperreflective dots can be seen somewhat in the color fundus photograph as a reflectivity, but it's not visible on autofluorescence, but it is visible on near infrared. The other feature on OCT is this hyperreflective line in the Brooks membrane. This is reflective on color fundus images, hypo-autofluorescent, but highly reflective on near infrared. So using 4 different imaging modalities, you are able to identify different features that might be important for future consideration. And in fact, this is becoming one of a new block, a kid on the block in terms of phenotyping age-related macular degeneration. So we asked the question, what are these made of, especially these what we call now nodules and these spherules compared to these plaques. We were lucky to work with people like Christine Curcio, who had the technology and the knowledge how to identify the very same structures in post-mortem eyes. When you take that, you have the opportunity to identify the exact structure when you are processing the tissues, and that allowed us to make sections of those nodule type structures which look like this on a normal pathological image. It certainly doesn't look something that you would expect to see in the eye. So the question really was what these are and what the function of these. It turns out when we use different methodologies, this is just one of them, X-ray diffraction, that these nodules are made of hydroxyapatite. Now, hydroxyapatite forms bone and hydroxyapatite forms your teeth. So why would you have a piece of teeth in the back of the eye? It's really a challenging question and a challenging concept to understand. It is until you start realizing what hydroxyapatite is, apart from forming bones and teeth. Those who remember their biochemical laboratory work might remember that hydroxyapatite is one of the most widely used chromatography column medium. And that means hydroxyapatite binds proteins and it's used to separate them for purification purposes. Is this something that can happen in the back of the eye? So here is an example of, this time I'm particularly concentrating on those little dots. When you look at looks for those dots on electron microscopy, if you look at the top image, you can see a healthy looking RPE, maybe a little start of a subclinical drusen with photoreceptor outer segments and the lumen of the choriocapillaris. If you zoom on these little dots, you see that they are fairly spherular, and in fact for these particular dots we identify them as about 1.3 micron in thickness. And what you see here is that the surface looks quite different. In fact, this is a beautiful image where there is this hydroxyapatite little spherules with a little afro hairstyle like appearance of something on the surfaces. When we stain these with different proteins, antibodies for different proteins that are actually present in drusen, like complement factor H or amyloid beta, what you see is that hydroxyapatite spherules do exactly what we thought it might do: bind on the surfaces these proteins and help the buildup of drusen. It's important also to mention that this hexagonal shape is the size of a single RPE cells. So you can see that you have multiples of these little spherules under RPE cells, and the release of material from these cells, depending on the site, will determine what binds to their surfaces. So based on this mechanism, we really developed this idea of drusen biogenesis, where in a normal state, this is electron microscopic and fluorescent imaging representation of this scheme, there is a transport of lots of things from the photoreceptors to the bloodstream, including lipids. And this is Christine Curcio's picture of lipid deposition in this interface. For some reason, these lipids can get stuck in this interface, and then for some reason, which we don't yet know, hydroxyapatites start developing on the surfaces, forming these little spherules. And we can see them as individuals, as you can see here. Once they are there, they will bind different proteins, and once they start, you will find how drusen form, proteins and lipids and different cellular junks will start sticking together to form drusen. So if we take this mechanism and now translate it back to the so-called nodules, which are very big, 100 micron or even bigger calcifications, similar to those little dots. If these can happen with these as well, what is the biological or clinical relevance of that? It turns out that a group in Los Angeles had a cohort of patients with this feature, and they showed that when these features are there, your chance of progressing to end-stage AMD increases by about 6.5–7 times. So it's very important. I want to emphasize this. It's not only that they can cause contribute to it, but they will contribute to a very rapid progression. So how diverse is hydroxyapatite deposition in the back of the eye? These are just two elements, zinc, which I used to work on before, and hydroxyapatite on flat mounts, and it's all these whitish appearances are hydroxyapatite deposition. So you have a lot of hydroxyapatite deposition in the back of the eye, and therefore there could be a lot of binding of different proteins, different lipids and so forth to build the resistance within the Bruch's membrane to block transport. So now that we identify these features, how could we move forward? There are several questions which we ask. Can imaging mineralization be used clinically for early detection, which is quite possible, but also for progression? What are the molecular steps involved in this mineralization? Because it's very unusual to find this in the back of the eye. So there could be systemic as well as local forces that contribute to it. And we need to understand that there is a link to genetics in this process. Does mineralization has a functional effect? And now I think it's becoming more and more accepted that it's a phenotype for age-related macular degeneration, but it's a big question whether there is a cellular function that these mineralization could affect, or whether how to use this. We were very lucky. Just recently we got a very significant funding from the National Institutes of Health for a program grant, and we have a team of different expertise assembled to answer many of these questions. So this 5-year program is just started recently. And here is just one example of where we could go. So this is fluorescent lifetime principle. Fluorescent lifetime ophthalmoscopy is now does exist, and we propose that that could be used to detect calcification in the human tissue. So what is lifetime? When you excite an electron, when it goes back to the stable state, it's not only emitting a photon, which we use as fluorescence, but it also has a lifetime, how long it takes to go back to baseline. It turns out that when we bind tetracycline or doxycycline to hydroxyapatite, the lifetime of the molecule changes very significantly from nanoseconds, from these different measures. They change very significantly in the presence when these tetracyclines bind to hydroxyapatite. And here is an example of drusen. This is the result of fluorescent lifetime imaging. You can see two fluorophores. If you highlight one of them, then you can see the appearance of the underlying retinal pigment epithelium. If you circle the other one, you can see that you can clearly distinguish hydroxyapatite containing drusen in the back of the eye. So there is one new methodology that we might be able to use in the future. And I'd like to finish here just with something that I think is quite interesting. The word druse is coming from German, and it's the name of this little geode in geology. When you look at our druse in biology, then you see this little similar crystallization within drusen in the human eye. So in 1815s, when this name was given to drusen in Germany, they didn't know what it's made of. They just looked at the appearance, and it turns out that they weren't really wrong. It looks really very similar to drusen.
Thank you very much, Professor Emre, for your informative presentation regarding the nature and ingredients of drusen, especially your last slide about the comparative comparison of the drusen in the geology and biology. As a veterinary surgeon, I had a different perspective on the drusen. What's your opinion on the significance of expanding our understanding of the ingredients of drusen and the natural progression of dry type AMD and the future treatments? As you are aware, current treatment options for AMD have centered on the complement pathway. What are your thoughts regarding the future of AMD treatment in this regard?
So if I want to be funny, then I would say that everything is about hydroxyapatite, but of course it's not. So in terms of how this fits into the future, I think understanding why these classifications start is part of the pathology, and therefore it could serve as a new target for the drug development. But it's also important to mention, since you asked about the complement system, that complement factor H, when it binds to the surfaces of this or any other complement proteins that it might be involved, they are very likely to change their function. And therefore, if they are not free to do their job, even if they are there, even if they are not mutated, they could be a significant problem. So stopping proteins to bind to these surfaces will help clearing them out, but it also should be able to help develop a better function for that interface. So I think the two things are quite separate, but there is a link between them. And if we were able to develop some kind of method of stopping this calcification growing, I think we will have an opportunity for better treatment.
Thank you.
Thank you very much, Dr. Lingile. Is there any other questions? Or we can go to Professor Ambrosio. I think there is no other questions. Next speaker is Professor Renato Ambrosio, which all of us know him by different helps, mainly in corneal image analysis and mainly on keratoconus diagnosis. He's a professor of ophthalmology in University of Rio de Janeiro and Sao Paulo. He has more than 300 publications, maybe much more than 300 publications, and received more than 50 international awards because of his great effect on ophthalmology. And in our daily practice, we can feel effect of his work in image analysis and especially in ectasia diagnosis. Let's go to hear his nice presentation.
Hello, I'm Renato Ambrosio from Rio de Janeiro, Brazil. I'm happy and grateful for participating in image analysis in ophthalmology. I thank Professor Hashimin and Professor Kodoparas for their honored invitation. My talk is on the Brazilian Artificial Intelligence Networking in Medicine. This is my financial disclosures, including Oculus, Brain AI solutions, and a masterclass online course with over 20 hours of presentations related to multimodal refractive imaging. I start with a quote. As for me, all I know is I know nothing, from Socrates. My humble interpretation is that the more that we know, the more that we should be sure that we should know more. So the idea is on the understanding of AI, and this is a path that we started about 15 years ago with my friend João Marcelo Lira introducing me to Professor Aidro Machado, who was then a young professor on computer science, just finished his PhD in Sorbonne University, France, on AI. So he taught us about AI, and we all learned tremendously from this over this last decade. And the idea is to get machines to help us. First, designing machines to do what humans can do. But now, what the capacity is the capacity to do things that we can't do. Looking at the multidimensional analysis is something that we cannot do, and machines can, but we have to be keen with strategic and understanding the whys and how to program those machines. We have diagnostics in refractive surgery. We have a lot of information to be used for decision, for optimizing, customizing, planning. If the surgery is going to be doing the cornea, phakic IOLs, or lens surgeries, a therapeutic procedure, LASIK, SMILE, and PRK. And we should decide that on the role for optimizing outcomes and preventing complications like ectasia, tear film dysfunction, epithelialization, quality of vision issues. And all this data are available. Topography, tomography, biomechanics, whole eye wavefront data. All this data should be integrated. And this integration would be key for us to make clinical decisions. The concept is that we have data acquired, parameters that are generated. We have to think about the precision, the repeatability, and the correctness of the value, considering a reference value, the accuracy. Those parameters are going to be used in clinical displays, and subjective analysis coming from here. Objective analysis from indices in artificial intelligence is what we should strive for. The algorithm A2I squared is the one I created to understand the application of an ancient intelligence, the philosophy, the whys, and application of artificial intelligence considering these whys and why and how. Considering ectasia after LASIK, the first descriptions in 1998 by Seiler, the first 3 cases with high myopia treated by LASIK, the impact from surgery caused a weakening in iatrogenic ectasia. A few months later, he published the paper on form-fused keratoconus. Here he defined form-fused keratoconus based on inferior steepening on axial curvature from placido-distal topography. Those 2 reports allude perfectly to the pathophysiology of ectasia. Biomechanical decompensation occurs considering the resistance of the cornea, innate biomechanical properties, and the impact from the environment. Any cornea may undergo the biomechanical decompensation, nicely described by B.J. Dupes and Cynthia Roberts, considering eye rubbing and the impact from surgery. Eye rubbing is the major cause, and if you want to avoid ectasia, tell patients not to rub the eye. Eye rubbing aggravates keratoconus or can even cause ectasia. So while any patient, any person on this planet would benefit from the information that eye rubbing is bad for you, I would say that any cornea surgeon, the more experienced they are, they should understand aggravating keratoconus and causing ectasia is not the same thing. This is a patient that I present many times. The residual stromal bed was 304, PTA was less than 0.4. The little yellow here on the pre-op would be pathognomonic, especially if you consider the ectasia result. But this was the initiation of a very important study that looked at the variability of subjective classifications on corneal topography. This is case number 6, varying from 0 to 3 with a mode of 0. 11 experts classified again, now from 0 to 4. The first classification was 1.5 diopter absolute scale with Steve Kleiss, Mike Smolik. Now with the holiday scale with a half a diopter normative, which is a classic ICs, 9 of the 11 experts had a different classification for the two scales. Looking at the pre-op tomography, we have Pentacam available from the U12 file. So the D was higher than 1.5. The arcmax was lower than 400. This is ectasia susceptibility. In a larger population, we collected 105 cases with a pre-op Pentacam data from patients that develop ectasia after LASIK, comparing to 3,278 stable cases. The D has a 59% sensitivity, much higher than the 22.9% sensitivity on the IS value. But we can go better with the D for the Pentacam random forest. This is the base for the BAD-D version 4, which will soon be available. And this is the work by Bernardo Lopez on artificial intelligence. Where are we? There's a very nice editorial that we put together. And this is important that we have to understand that those cases down here make the point that we should still continue to evolve. Age, PTA, and residual stromal bed in this population does not explain so well what happened. So we need to go beyond, and that's the work on the relational tissue altered, which is an AI characterization of the impact from laser vision correction. This is the second thesis of PhD that Ido Machado is doing, and I'm honored to collaborate with him on that. So he looked at the solutions for imbalanced groups, much larger population on this table, and he was able to segment the population and do strategic analysis so that we were able to improve statistically significant analysis with the relational tissue altered along with the BAD-D today. So this is a very promising work that we have been implementing on clinical practice still today. Ectasia susceptibility is what we should look for beyond, not over topography and tomography. Biomechanical assessment is fundamental. The Oculus Corvis ST is a tonometer that gives us that deformation response on Scheimpflug imaging. So in 31 milliseconds, we have 140 frames that characterize the cornea, so a keratoconus index can be created. Nice work by Ricardo Vinciguerra and Paulo Vinciguerra that got the Troutman Award in 2017, in which we are able to characterize if the cornea is softer or stiffer. The integration of data from tomography and biomechanics has been our work since 15 years ago. I started with the ocular response analyzer, now with the Corvis, and not only with biomechanical data, but also with data from very asymmetric ectasia cases. So in this work, published in 2017, we have 94 cases with normal topography in which the fellow eye had ectasia. Normal topography is defined as a strict criteria, and we had 90.4% sensitivity. We improved that in terms of the AOS thesis, which goes for 551 normal topography eyes. And this is going to be on the analysis, a very important path, so that we improve the sensitivity for the very symmetric normal topography cases with about 10% increment, with 84.4% sensitivity. We can optimize it further with ethnicity and also multimodal refractive imaging like epithelial thickness data. The Brain is a software that is available online, braincornea.com, since 2015. We integrate data along with the available pre-op data that you have, Pentacam, Oculizer, keratograph, or general topography. We are expanding that for a commercial software that will be released soon. And this year, we were honored to collaborate with the JCRS in this consultation section in this 27 years old lady that had low myopia, and we were able to identify ectasia susceptibility very objectively. So the idea is the understanding if the patient is a good candidate for lamellar procedure, LASIK, or SMILE. There's an advantage of SMILE for LASIK, but not for PRK, surface ablation. You have ICLs, phakic IOLs, ICRIL-phakic, and others that are coming to the market that are very robust and very trustable with excellent results, or if the patient is a good candidate for therapeutic corrections. So the relational tissue alter will be, along with the TBI, the way to characterize this best procedure for the patient. So we apply artificial intelligence with ancient intelligence. We are an entrepreneurial company, not academic center anymore. And the solutions that we are providing with ectasia susceptibility, with an enhanced ability for making this as the best as possible for helping our patients using refractive surgery the best way as possible. So thank you very much for your kind attention, and I'm honored to collaborate with you and eventually have some question and answers very soon. Thank you.
Thank you very much, Professor Ambrosio. As in daily practice, we feel the effect of your work in or practice. The main thing I just get familiar with the artificial intelligence, it's coronary refractive surgery, and I now can feel the effect in my daily practice. Next, dear speaker is Professor Kafi. He's assistant professor in bioengineering at Durham University. Her research is focused on artificial intelligence for biomedical image and signal processing. Through close collaborations with the clinicians and NHS, she has long experience in medical data processing using artificial intelligence. She has published over 120 publications in especially this artificial intelligence field. And we are eager to hear your nice presentation, Professor Kafi.
Hi there, thank you very much for your invitation. And do you have my screen shared?
Yes, we can see your screen and hear your voice.
Fantastic, thank you very much. Well, I would like to talk about the application of artificial intelligence in ophthalmic images. However, I would like to concentrate more on neurodegenerative diseases, discrimination of which is somehow more complicated rather than the easier complications in ophthalmologic diseases. Neurodegenerative diseases actually talk about a kind of pathology as a group of disorders that yield progressive degeneration of the nervous system. It includes diseases like Alzheimer's, Parkinson's, PDD, Lewy body, multiple sclerosis, and NMO. We all know that what happens in diseases like Alzheimer's, for example, a kind of shrinkage happens in the brain, or we know what happens in Parkinson's. A particular part of the brain doesn't work properly, and then we don't have enough dopamine. And over the time, the cells will be lost, and lots of symptoms like dizziness, trouble in moving, and so happens in Parkinson's disease. We have very similar symptoms in other neurodegenerative diseases, but the case is that the changes in the brain are usually difficult to detect, and there might be already damages even before the symptoms would appear. So everybody is after a kind of way to detect the neurodegenerative diseases in their earliest stages. Somewhere that we can concentrate on rather than the brain is the eye, I mean the retina. Actually, retina is the outgrowth of the brain, and it's the only part of the central nervous system that can be seen directly from the outside. So we may use many ocular imaging modalities in order to see what's happening inside the retina and somehow see it as a kind of mirror to our brain. Ocular imaging modalities vary And you know, there are lots of them, like 2D images of the eye, like fundus images, and 3D data like optical coherence tomography gathered from macular region or around the optic nerve head, like peripapillary OCTs. So optical coherence tomography provides a kind of 3D information for us to see what's going inside the retinal layers. So the fact that OCT is useful in diagnosis of the neurodegenerative diseases is completely known. There are lots of papers discussing what happened, like loss of the retinal layer, fiber layer, and ganglion cells in Alzheimer's disease, or what happens in inner retinal layers, specifically the cells that might contain somehow dopamine, and their study in Parkinson's disease is also published in many works. We are also aware of the studies that show the ganglion cell layer and plexiform layer get somehow thinner in patients with multiple sclerosis. So the case is that in healthy people compared to people with NDD, we would like to see what happens in their OCT data. The reality is that the differences are really subtle, and this is the reason that I wanted to talk about neurodegenerative diseases rather than the ophthalmologic ones. You know that the changes in, for example, AMD or DME are completely obvious when you take a look into the OCTs. However, in neurodegenerative diseases, like the ones that I'm showing here for Parkinson's, the changes are really subtle and are not seeable by experts, by ophthalmologists, anybody. So what we can do, the case is that we need to somehow quantify the values, compare them to normal population, and find if there is a kind of change due to neurodegenerative diseases. The main aspect in this area is finding the location of the retinal layers in OCT images, which is called segmentation of the retinal tissue layers. All of you, when did a kind of segmentation step, know that it is not an easy one if you do it manually. So it's very time consuming, prone to error, and the differences are really small, and people get tired of doing so. So what we want to do in the first step using artificial intelligence is having a kind of segmentation and dealing with lots of other issues regarding such data, like working with large volume of data, doing quality control, as I told you, segmentation, and finally doing a kind of classification. So this is the overall aim. We want to start with OCT data, do a segmentation, and then finally do the classification. Well, we are aware that there are lots of segmentation algorithms already developed, and we may find them on devices like Heidelberg, Topcon, everything that we're using. But the case is that they are not reliable for every single data. For example, if you're gathering a lot of data for your research coming from different devices, it cannot work on all of them. And particularly if you're interested in working on neurodegenerative diseases like Parkinson's, Alzheimer's, multiple sclerosis, when the layers are really thin, the results of the segmentation with device is not reliable. So what we want to do is designing a kind of AI algorithm for segmentation of the retinal layers in neurodegenerative diseases in a way that is completely reliable and generalizable. So for this purpose, we start with training our AI algorithm, and then, well, the main aspect is finding a lot of data that's very necessary to train a good AI algorithm. So the data collection is done using a lot of data with demographic data, and also the ocular images like optical coherence tomography, fundus, OCTA, and so. So we have a very good database, and then we want to do a kind of segmentation. First of all, we developed a kind of tool, a web-based tool that gathered the manual segmentation in a limited number of the data done by ophthalmologists. And the process was watched by our AI algorithm. I mean, the AI algorithm is looking into the manual segmentation and learns how it works by its process. And then finally, we have a learned and trained algorithm that from now on can be used on any new data. And we can expect the segmentation of the retinal boundaries in 2D and 3D would be achieved. So such segmented retinal layers can be used for our next step algorithms. Just to take a look at what these information bring for us, we should just somehow compare the location of the retinal boundaries with what we have inside our eye. And it's a simple comparison, and you can see that the distance between mutual pair of the retinal boundaries is exactly somehow showing the data and the number of cells and the population of the cells in each layer. So if we calculate these distances and show them as a kind of thickness map, we can say that these colors are coding the amount of the retinal cells in each area. Accordingly, such thickness maps, and if we concentrate on 8 of them, for example, are a kind of low-dimensional and most relevant representation of the 3D OCT data. I mean, from now on, instead of sending a bulky 3D OCT data into our algorithms, we are able to just send 8 or even less number of the layers, depending on the disease that we are discussing, and looking into previous publications in clinics to see which layers have the most important effect on the output, and selecting such layers, just 1 or 2 or 3 of these images, to be sent into our next step algorithms. So if we take a look at the whole process, we gathered a lot of data, we trained our segmentation algorithm, and then we found these 8 thickness maps in order to be sure that the segmentation is doing properly. We compared the location of the retinal boundaries with what the expert clinicians have done for us. So we measured the kind of success rate in our segmentation. And in the next step, we want to do a kind of classification. I mean, we want to send such retinal thickness maps into an AI algorithm in order to say if somebody is suffering from any specific neurodegenerative diseases. So in this step, similarly, we have neurologists who label the data, actually having a lot of other information like brain MRIs and blood tests and so. So we have the label for our images, and again, our algorithm watches and learns the process. And finally, we have a trained algorithm. And from now on, we expect just to send the retinal thickness layers into our algorithm, and the algorithm gives us a kind of answer if the person is suffering from any specific neurodegenerative disease or is healthy. So this is the overall aim. And similarly, in order to measure the success rate of the classification, we again compare the performance of our algorithm with the expert clinicians. So what are the challenges that we had and how we just found the solution for them? The first one is the reliable automatic segmentation, as I told you, and the case is that we have a lot of people with different neurodegenerative diseases. Therefore, they might have some atrophic layers, some indistinct boundaries, some media opacity, or artifacts due to movement, particularly if we are talking about Parkinson's. And also the images are coming from different devices like Topcon, Heidelberg, Zeiss, so. And the imaging protocols vary, and the resolutions are different, size, artifacts, everything. And you can take a look into this bunch of the data to see what kind of variety we are talking about. So we prepared a kind of a segmentation algorithm that is very robust to all of these changes. It is called NDD-seg, and it finds the boundaries in many different steps. In first step, it finds the location of the retina, discriminates it from the background. Then we called it RTSN. And then it concentrates on interretinal layers to find the exact location of the retinal layers. So in order to do this job, finding exact location, we also incorporated the information coming from the texture of the layers, because what we actually do with our eyes is just find the kind of change between the texture and find which one is similar to RNFL and which texture is similar to GCIPL, the ones that we remember. So we try to do the same process for our algorithm. It learns the texture by seeing the many samples. The other thing that we did was that most of the segmentation algorithms designed automatically have the kind of measurement to say if the location of the area that we're looking for is found correctly or not, somehow finding kind of overlap between the mask and the one that we are looking for. However, The fact is that in segmentation of the retinal layers, rather than just talking about the areas, the location of the boundaries and exact location is really important. So we added a new segmentation loss. It is called a boundary preservation loss. And rather than just seeing the overlap of the areas, we also looked into exact locations to be very precise. We used a bunch of data for training and testing. The initial 111 OCT volumes, each coming with 128 B scans were used from 90 healthy controls and 21 patients with multiple sclerosis. So we used 88 of OCT volumes for training and 23 randomly selected ones for testing. And finally, after training and testing our algorithm, the algorithm is now trained, and we want to see how it works for predicting unseen data. For this purpose, this time we didn't use the same bunch of data that we trained our algorithm. It was from Iran. Instead, we just tested it on different data sets from the UK, coming with different NDDs and from different locations. You can see that the bunch of the neurodegenerative diseases that they were suffering from, and also the types of the devices that we used, and you can see the segmentation results that are satisfactory. If we compare it with the other algorithms, like a very simple one like UNET, you can see that the performance in the lowest row is relatively better than what you can find with easy segmentation methods. And we also compared the performance with what the devices are gathering for us, for example, the Heidelberg segmentation. And the fact is that if the person is suffering from a specific disease, or if the quality of the data is so low, our segmentation algorithm surpasses in performance compared to the devices like Topkan and Heidelberg. So after finishing the segmentation algorithm and finding those 8 retinal layers, thickness maps, we wanted to do a kind of classification to say which person is healthy or which one has a kind of NDD. So we concentrated on multiple sclerosis. We used the eyes of healthy people and multiple sclerosis eyes. So we used 2 different data sets, one free data set available for public use by Baltimore, and another one that we collected in Isfahan with 24 healthy controls and 60 multiple sclerosis eyes. So we used different classification algorithms. First of all, because the data set was not that big, we concentrated on a kind of feature extraction algorithm. So we use autoencoders, and in this way, we show the retinal thickness maps of 3 main layers, RNFL, GCIPL, and INL, as input, and also the output of the autoencoder. And we expect that autoencoder can somehow mirror the data. And when it does it perfectly, we can come to conclusion that the middle part, which is called the kind of bottleneck of our autoencoder, has the needed information as a kind of extracted information in it. So this shallow neural network used after this step applied on the latent layer information coming from those button of the autoencoder, and we did the classification using the shallow neural network. In another work, we used the CNNs from scratch. We just showed the data from 3 main layers, and we just wanted a 2-class classification as out of our CNN. As you know, that when we design a kind of neural network, there are lots of parameters to consider. So we somehow modified them and found the best combination of the layers to do the job for us. And finally, there are some works like they're called like transfer learning. And in such works, we usually have a kind of network that is trained, pre-trained on a data that's not essentially the data that you are working on. For example, you may have some networks that are trained to distinguish cats from dogs. There's no relation to eye. However, the fact is that such algorithms can be used by a kind of transfer learning and fine-tuning into our data. And the reality is that the neural networks usually have many different layers. The first layers extract a very general information that is very similar, even if you're talking about cats, dogs, or medical data. So if you concentrate on those layers and use the same information coming from such pre-trained algorithms, you only need to train the final, very fine information coming from our medical data. And this is what we did with different pre-trained networks, including ResNet. Finally, comparing all these information, autoencoder, Scratch CNN, and also pre-trained networks, we found that our Scratch CNN has the best performance, and using the thickness map of 3 main retinal layers, the accuracy of 97% was achieved. Something that's also important is just to see if the algorithm can be generalized in another data set, because it's very common that you just concentrate on a kind of data, get good results. However, if you change it and take another data into it, the performance goes very bad. So it's really important just to see if you train an algorithm on this data, how is the performance on another data, on seen validation data. So we did the same task. We trained on the data gathered in Isfahan and just wanted to see how's the performance on Baltimore dataset. And you can see the accuracy is still acceptable, like 84%. The other important aspect in working with AI is the fact that nobody likes a kind of black box. And this is the kind of something that everybody is suffering. When we talk about AI, everybody doesn't like the way that it just says it is healthy, it is coming from a disease background. Everybody would like to say, how did it come to this conclusion? And this is the fact that we talk about interpretability. There are many methods to say how my algorithm is working and which area of the image my algorithm is looking into. So which area has the most important information. And there are many different methods for this purpose, like occlusion methods and GradCam methods. We used many of them. And these are the results, as you can see. It shows that, for example, the area around the fovea in GCIPL has one of the most important areas. Lots of information for distinguishing multiple sclerosis from healthy people. And the fact is that these outcomes were completely similar with what the clinicians would expect. I mean, the combination of neurologists and ophthalmologists come to conclusion that statistically talking, these information are really important. So we had the very similar outputs using different methods of AI. And we can say that our algorithm looks exactly to something that it should look. For example, you can imagine that there is a kind of tiny text in a corner of your data, and you may find finally that the algorithm doesn't look into your data, it's just looking into that text. It's a kind of cheating. So it's important to say, is it looking into the same area that the clinicians would concentrate on? And this was the fact that, yes, it is looking. So overall talking, this was the whole story. We had some OCT scans, we did segmentation and found the retinal thickness maps and did the classification and found the probability of having different neurodegenerative diseases. I had just a report of multiple sclerosis. We are working on many different data coming from Parkinson's, Alzheimer's, and so on. The whole process can help us in the referral decision. So talking about the benefits for patients and public, we can say that the algorithm is useful in early diagnosis and early intervention, and also the contribution to AI. And the benefits to NHS and the community clinicians is the optimization of the clinical workload, reduced cost of screening, and streamlining the referral process. Thank you very much for your attention. And I would like to invite you to take a look into my website if you're interested in seeing many codes and also available softwares, anything that you might find somehow useful if you're interested in implementing any codes in this area. Thank you very much, and I'm happy to answer any questions.
Professor Kafiye, it is a pleasure to meet you again.
Thank you very much, me too.
I greatly appreciate your presentation. In Farabi Hospital, we are honored to conduct joint research with Professor Kafiye, and it's a kind of honor for us.
It's an honor for me. Thank you very much.
Thank you. Thank you. Actually, I haven't any question, but sometimes I imagine that in future, there will be some stations in cities that can capture images of your eye and predict the likelihood of future diseases like Parkinson and other neurodegenerative disease. This is both incredibly fascinating and also terrifying. I don't like to be aware of the high risk of Parkinson or Alzheimer in future. So this is the scary side of artificial intelligence that in future that will implement in the clinical practice.
Well, I exactly agree with you, but the fact is that I think that we should accept that the positive side of this fact is really high, because nowadays the new lots of new drugs are already accepted by the FDA and any other certificating companies to say that the early diagnosis can help the early treatment and help better life for people with Alzheimer's, Parkinson's, and so. And also, if I myself would know that I'm going to suffer from Alzheimer's, I might change the style of my life, and this can somehow help the lower actually process of suffering from the disease in long term. So I believe that it's not that scary.
Thank you very much, Professor.
I think Dr. Ringil has some questions and points to add. Would you please unmute him?
Professor Imre, can you ask your question, or on behalf of you, I can ask your question. The Professor Imre asked that, do outer retinal layers similarly well segmented by the AI approach?
Well, we did the segmentation both on the data from fovea and also peripapillary data around optic nerve head. Yes, and we found that the location of the boundaries are really being segmented precisely compared to what we have with our devices.
Thank you. One of the audience, Farah, is now raised the hand. You can unmute yourself and ask your question, please.
Thank you so much. Professor, I have a question. You mentioned that in AR, you're using the same algorithm that can differentiate the dog and the cat, and you're using the same thing for the normative images. My question is, would you please give us some more details? Because in neuroimaging, there are very, very small subtle changes. But for example, the algorithm that is used for dogs and cats are so huge. So would you please give me some more detail about this?
Yeah, sure. Well, it's just a kind of sample to somehow simplify what I wanted to say. But the fact is that, yes, when we're talking about different layers of a kind of algorithm like convolutional neural network or CNNs, the fact is that the very first layers just extract very rough information. Let me say like the lines or the spots or just kind of noise in background. But when we come into the last layers, the information in the layers and what we can see from the vision maps is very similar to what we are really interested in. I mean, if we are talking about cats and dogs, their shape goes like eyes, ears or so. But when we are talking about the eyes, they become similar to vessels, to the location of the fovea and so. So what happens if we want to use a kind of algorithm developed for cats and dogs and then use it for our real algorithms in medical images? The fact is that our networks are very, very big. Like, for example, you may have 10 million numbers in them that should be trained. But the fact is that usually when we are talking about the medical data, we don't have a lot of data. So sometimes it's not that easy to train an algorithm with lots of parameters like 10 million, just using 100 images. So what we can do is first train our algorithms using cats and dogs. So the fact is that the lines, the edges, the spots are similar. There is no difference. So a part of the network that is just gathering such information like lines, edges or so can be used. And we just freeze those information from 10 million data that we wanted to train. Let me say, I could just freeze like 6 million of them. And I only want to train the rest of those 4 millions. So there's a reduction in the number of parameters that you want to train, and you might be able just to train those parameters using a small number of data. So you're not using the cat and dog network for your classification algorithm. You're just freezing a part of data and then you showing your real data and training the rest of the network with your real data. Therefore, you can expect that the algorithm is working with your somehow very different data.
So in this way, can we get a precise information and based on the things that you explained, is the final result will be precise?
Yes, yes. You can take a look into the words like transfer learning or fine tuning in deep learning, and you can see, yes, the results are completely reliable and good, because the fact is that the important part of the network is being trained with your real data, your medical data, and you're just taking advantage of those information that are common between cats and dogs and the medical data. So it doesn't matter if you're using the first data coming from animals, from satellite or anything. It's important just to concentrate on a part of data that you are gathering and a part of network that you're training that with it.
Thank you very much, Professor Kafiye, for your very nice presentation. We hope to have future cooperations with you and Farabi Hospital together.
Thank you. It's an honor for me, for sure.
I just saw that Professor Ambrosio is online. Can we have him? He's not online again. Professor Ambrosio, are you online? I just want to ask Professor Aksaifard, is Professor Aksaifard online? Yes. I just want to introduce Professor Aksaifard as an ophthalmologist. I have many times had problem diagnosis of optic nerve diseases. And if we can find an artificial intelligence software or device to help us diagnose optic nerve diseases, it will be interesting for me, at least. Professor Aksaifard is Professor of Ophthalmology in Farabi Hospital, Tehran University, who has many high-quality publications and has mainly worked on image analysis in diagnosis of optic nerve diseases. We will hear his speak about his experiences in optic nerve image analysis. Dear Masoud, are you online? I think there is a problem with the voice.
Hello.
I think it's better.
Hello.
Hello, hello. We can hear your voice.
Oh, hello. Thank you, Hassan, for your nice introducing me. If I want to categorize doing my research in this field, I first mentioned that it is started about 8 years ago that I started using OCT angiography images for diagnosing different optic neuropathies. In that times, all OCT angiography images had a lot of artifacts and a lot of redundant information in the images, they had a lot of shadows. And then we decided to remove those shadows and those redundant areas to get better information from those images to differentiate between several optic neuropathies, including glaucoma and ischemic optic neuropathies. And at that time, I got several helps from several centers, especially from Dr. Jalili, who is now in San Diego working on AI. And then I started to use some fundus photographs to differentiate several different optic neuropathies, including glaucoma and non-glaucomatous optic neuropathies. And then I got several help from Dr. Kafie, which had a very nice presentation before me. So now I think we need to do a lot in this pathway, and maybe in the future we could diagnose optic nerve disease easily with AI. And now I believe that AI is much better than me, because we did a recent study and we found that AI is better than expert specialists for differentiating glaucomatous and non-glaucomatous optic neuropathies. And also, I did collaborated with one international study, and that study differentiated papilledema from other optic neuropathies. And again, AI was better than several expert neuro-ophthalmologists. So I think in the future, we need to stay at home, and then the AI will diagnose every optic neuropathy.
Thank you very much, Masood. So hopefully with your future works, we will have an add-on on maybe OCT to tell us that the exact diagnosis of the patient and will help us for the diagnosis. Thank you very much. Masood was in a journey, and I asked him today to do a short presentation. We were honored to hear your nice experience. Thank you, Masood.
Thank you for you.
Thank you. I'm honored to introduce Professor Khalilpour, who is a faculty member of Farabi Hospital. All of us know him, and he helped us for this webinar too much. He has many high-quality publications in vitro retina field, and many of them are the field of image analysis. We will hear his presentation and about his experiences on image processing in retinal diseases. We are waiting to hear your presentation, Elias.
Thank you, Professor Hashemian, for introducing me. Can you hear my voice and see the presentation?
Yes, yes. The voice and the slides are great.
Thank you. Honored guest and fellow enthusiast of ophthalmic imaging and image processing, I am absolutely delighted to welcome you to this fascinating discussion on a field that has completely transformed our understanding and diagnosis of eye conditions. I do not have any financial interest related to the subjects that will be discussed during this session. The human eye, often called the window to the soul. The eye is not just a complex and carefully constructed organ, but also a remarkable example of the wonders of modern technology and medical science. Ophthalmic imaging gives us the ability to look through this window with exceptional clarity and accuracy, allowing for early identification, diagnosis, and tracking of eye disease. Ophthalmic imaging is a rapidly evolving field that is constantly changing, and one of its major driving forces is image processing. This digital image technology enhances the raw data collected from imaging devices, extracting meaningful information and facilitating accurate diagnosis and treatment plans. Image processing in ophthalmology can be classified into the following areas: pre-processing, feature extraction, segmentation, and classification. Let's take a look at examples of each subclassifications of the image processing in retinal disease. Fundus photography, OCT, and fluorescein angiography, and new imaging modalities like OCTA are just a few of the imaging techniques used by physicians to get a good look at the retina. Processing is subsequently applied to these images to enhance their quality and draw out useful data. Pre-processing step can be categorized into the following sections: noise reduction, contrast enhancement, image registration, segmentation, artifact removal, color standardization, and data augmentation. Let's review certain examples of them. Images of the retina are susceptible to noises, which may be caused by camera sensors, illumination conditions, or patient eye movement. Filtering and other image pre-processing techniques can help reduce noises and improve the clarity of the retinal images. Adjusting the contrast of the retinal images can enhance the visibility of essential elements such as blood vessels, lesions, and other abnormalities. Contrast enhancement techniques improve overall image quality and facilitate the identification and analysis of retinal conditions by medical professionals. As you can see here, the hemorrhages inside the retina can be clearly visualized after a contrast enhancement. Segmentation is another method of pre-processing, is an essential step that identifies and isolates regions of interest like intraretinal or subretinal fluids. For instance, it can be utilized to outline the borders of blood vessels, lesions, and optic nerve head. For diagnostic and monitoring purposes, segmentation algorithms aid in quantifying and measuring these structures. In a published study conducted under the guidance of Dr. Kafieh, we tried to develop a semi-automated algorithm capable of accurately segmenting the retinal layers and intraretinal fluids in OCT cross-sections of diabetic patients. Even in patients with severe macular edema due to diabetic retinopathy, this algorithm works precisely. As you are aware, the manufacturer's built-in software makes some errors in segmentation of the retinal layers. So the methods, automated or semi-automated methods that developing every day, that can be very helpful for ophthalmologists to monitor the progression of disease. Images of the retina can include artifacts, as the Dr. Aksaifer addressed. These artifacts such as dust particles, fringe shadows, and reflections. Image pre-processing techniques can detect and eliminate these artifacts, ensuring that the images are free from distractions that may impact the diagnosis. Data augmentation techniques can be employed at the pre-processing stage in machine learning applications for the diagnosis of retinal disease in order to enhance the variety of training data set. This is helpful in enhancing the strength and applicability of the machine learning models. As you can see in this picture, by rotating the fundus images, you can augment your data set. The process of feature extraction plays a crucial role in the evaluation of retinal images for the purpose of diagnosing and evaluating retinal disorders. The process entails the identification and quantification of pertinent features or patterns present within the retinal images. The collected features are utilized as input data for the range of machine learning algorithms, diagnostic tools, and decision support systems. And the followings are the essential functions of the feature extraction in the context of retinal diseases. The process of feature extraction plays a crucial role in the identification of anomalous structures or patterns like drusen or exudates or hemorrhages. And in patients that are diabetic or in patients with age-related macular degeneration, this feature extraction can be very helpful, even in diagnosing and also in the monitoring of the disease progression. Specific clinical indicators such as retinal thickness measurements, optic cup-to-disc ratio, or blood vessel diameters can be quantified by extracting relevant features. The acquisition of these measures is crucial in order to monitor the advancements of the disease and evaluate the efficacy of therapy. In this article published in the BMC Medical Imaging, we described the development of an automated machine learning algorithm for segmenting two hyperreflective retinal layers, IPL and OPL, in OCT images. The algorithm uses a classical SVM, support vector machine regression methods, alongside a wavelet kernel, and calculates the smoothness of the retinal layers. In this study, we compared the automated segmentation with manual segmentation by experts and showed the modest discrepancies. And finally, we concluded that the algorithm can be a reliable and convenient approach for segmenting the IPL and OPL, and also for calculating the smoothness of these hyperreflective retinal layers in OCT images. Following the aforementioned investigation, our objective was to evaluate the precision of our algorithm in distinguishing between diabetic patients with varying degrees of severity. For instance, we conducted a comparative analysis for retinal layer thickness measurements in nasal, temporal, and also in the superior and inferior sides of the fovea. And our findings demonstrated that a smoothness index of the inner plexiform layer in the nasal area can precisely differentiate severe NPDR patients from PDR patients with the measured accuracy of about 95%. In a separate study, we demonstrated a completely automated method for calculating the CVI measurement, choroidal vascularity index, in the OCT images. This study was conducted under supervision of the Professor Kafieh, and by utilizing the proposed unit model, we could segment precisely the choroidal boundary and also the measurements and calculating the choroidal vascularity index in the OCT images. Extracted features provide quantitative data that can be used for objective disease progression monitoring and evaluation. This is particularly crucial for the monitoring of chronic retinal disease like neovascular AMD and diabetic retinopathy. In a separate study that we honored to work alongside Dr. Kafieh, we present an algorithm based on the artificial intelligence for automated segmentation of CNV areas, choroidal neovascularization areas, and identification of CNV activity criteria in OCT images. The algorithm consists of two steps: pre-processing and segmentation of the CNV, utilizing a modified unit network, and binary classification of 5 activity criteria utilizing multiple models. The accuracy of the proposed algorithm in segmenting CNV and classifying the activity criteria was high, and the study indicated that the AI-based algorithm can detect and segment CNV from OCT images without the need for contrast agents, enabling objective and repeatable evaluation of CNV characteristics. And finally, the challenges and limitations in image processing for retinal diseases are image quality and interpretation variability, data privacy and security, and computational complexity that can be addressed in future studies. And future prospects and advancements in image processing for retinal disease can be powered by use of artificial intelligence, the remote retinal image analysis by the telemedicine approaches for timely diagnosis of the patients, and image-guided interventions. In conclusion, I would like to express my sincerest gratitude to everyone who is present today. Together, we embarked on an expedition to explore the fascinating field of ophthalmic imaging and image processing. Our goal was to discover the marvels of the human eye and cutting-edge technologies that enables us to gain a deeper understanding and provide better care for this priceless gift of the nature. Thank you very much.
Thank you, Professor Khalilipur, for your nice presentations and nice ending. I just want to thank all participants and all their keynote lecturers. I hope to feel the effect of image analysis in our future ophthalmology practice. Have a nice time and goodbye. Hope to see you again. Excuse me, I should thank Mr. Omidwar and Ms. Sadeghi for their great work in preparing this meeting. This was a group work. Have a nice time.
Thank you very much, Professor Hashemian, Professor Tandepeto, Professor Kafiye, and Professor Imre. Thank you very much.
Goodbye.